Hello again everyone. Now we'll talk more details about coupled and other programs that can be used in multiple sequence alignment. In coupled, it can be found easily in NCBI. You can download your sequences and of course paste it in coupled. Coupled is a very good and strong program and also can align and produce multiple sequence alignment that is fairly accurate. And of course this is embedded with MSA viewer and MSAV, that's what they call it. You can find it in the top here, on the top of the result. In this MSAV or MSA viewer, you can see many. You can get a lot of information from your alignment. Also, the MSA viewer can calculate the phylogenetic tree for you. If you just even put the mouse over the top bar, you can get even where is the position of this of the nucleotide in the sequence, and also the position of the nucleotide in the alignment. Of course, you can enlarge and see this in more details if you just enlarge the view from this bar. Also, you can see the alignment parameters in that. We will come later to the MSA viewer in the next lecture. Anyway, so you can get your alignment, multiple sequence alignment, in the FASTA format or even other format. You can realign your sequence. So coupled is a very good program that can be used for aligning your protein sequences. Cluster omega is another one. Cluster omega can be found easily in EPI. Cluster omega is, as we said, is you can choose DNA or protein to align. You can just paste your sequence and then submit, and you can get your result. You can color your result. You can download the file, and also you can get phylogenetic tree if you want, based on the multiple sequence alignment. The third one is TCAFE. TCAFE is more accurate. It has a lot of program inside it, which can be used and with different flavors. So you can do RNA. There's some specialized for DNA, some specialized for protein, some specialized for RNA. So you can use TCAFE or MCAFE depending on what you are looking for. TCAFE and MCAFE usually align. It can work with DNA, RNA, or protein using default parameters. So for RNA alignment, for example, you can use RCAFE and SARACAFE. And both of them are different. One of them align RNA sequences using predicted second structure. The second can align RNA sequence using tertiary structure. But both of them are specialized only for RNA. Protein alignment. There is many flavors in TCAFE that can be used for protein alignment. For example, Expresso can align protein sequence using structural information. Also, you can PSI-CAFE align distantly related protein using homology extension. So that's why if you have a problem with your sequences, which is could be distantly related or something, just you can use PSI-CAFE. You can overcome the problem of distant protein. TMCAFE is specialized for transmembrane proteins. So if you have a transmembrane proteins and you're working with this type of proteins, so TMCAFE is the good option for you. ProCAFE actually for DNA, for the aligned homologous promoter region. So it works only in the promoter region. So aligning them and extract good information from them can be done with ProCAFE. Accurate is combine most accurate modes for DNA, RNA, and protein, and combine is a very good one that can be used for combining two or more multiple sequence alignment into a single one. So you can easily, if you have two or more multiple sequence alignment, you made them by other programs, for example, or even you made them by TCAFE, you can simply combine these two or more multiple sequence alignment files in combine. Evaluation. There is many ways to evaluate your multiple sequence alignment using TCAFE. Simply, there is a core to evaluate your alignment output, and a program called IRMSD ABGB. It's another one also. And TRMSED, it's allowed. This also can evaluate your sequence. And strike, of course, can evaluate your protein in multiple sequence alignment in single 3D structure. Advanced, there's many advanced options. Actually, you can change the advanced options beyond this lecture. So and simply you can get your lecture and you can get your result and see the result of your alignment easily by colorful way. So and there is a color scheme here that can help you to interpret and help you to see the most conserved places, the most aligned places, and the other places that is not fairly aligned with TCAFE. There is many useful link and mirror sites for TCAFE, so you can take a look at the website and you can see that it's a very important tool that can be used in bioinformatics. Of course, if you have a large set of data of sequences, there is a program for that, which is Muscle. Muscle is a very strong program. As we said before, you can find it in the official website of Muscle, which is Drive 5, and you can find it also. You can find Muscle also in the ABI MDL website. So you can just crunch if you have several hundred of sequences and you would like to have a very strong program that can deal with this number of sequences, you simply can use Muscle. Muscle can be found easily, as we said, in MBL API website, and it's a matter of pasting your sequence, copy and paste your sequence in the website, and then it will take maybe a little bit of time, but after that will give you the results that can be used in other, if you would like to see it with other viewer or something, multiple sequence alignment viewer or something. In your own time, you can pick up your set of sequences, and try to find the differences between these four programs. You can see the difference in time, the difference in coloring, the difference in information that they can produce, which one is better for you, which one is really good for your experiment, and which one you can get more information from. Thank you, and let's go to the next lecture.